like this is focused on, presentation is focused on the audience, those who have some role in research writing and protocol writing. They Every individual who works in clinical research actually needs to analyze research protocol or some part of it so that they can perform their functions properly. Everything that is all studies conducted in the industry, they actually have a written protocol on how to go about it, and we'll touch all aspects of it as we go along. Um, so there is difference between writing versus analyzing. The analyzing research protocol, like different sections of it, people have to do in order to understand what needs to be done. And One person can do a whole analysis of protocol. The idea is to see that protocol is actually defining the research question and how you get answer for that. Is it like the design is good? So it helps out identifies any weaknesses or gaps in the protocol. For example, if the sample size is not good enough or great, so the power of the study is not good, so we need to put more patients. Or if we did not calculate the number of participants who might drop out of the study, then there is going to be a problem in when we analyze the results in the end, that it is good or not, we can prove the null hypothesis or not. The writing protocol involves a whole team. No one person can do that. It involves a creation of a detailed plan for a research study, and you have to clearly define the research question, the objectives, what kind of study design you're going to use, what methods you're going to use, how you're going to collect data, what are the ethical considerations for participants? Are we going to involve vulnerable people? Are we going to involve what age group, what disease criteria and conditions? And so it involves like the statisticians, it involves people from ethics. You have to get approval from ethics board, and the research scientists who have the background. And so there's so many different people who sit on the team who write protocol, and each one of them writes their own section, and it is reviewed by the person. There is a steering committee who sees how things are going, and then they go on like editing it. There are protocol amendments if something is not working, and new things come up when they implement the protocol in real world. So we look at the various sections of how to analyze the research protocol. So first step is to understand the research question, the primary question the research is seeking to answer. So we're looking for a clear and well-defined research hypothesis. That should be specific, measurable, achievable, realistic, and time-bound. Then we look at the next section is the literature review, that whatever has been done in that section, in that part of the like science till now, are there any gaps which are present? Like for example, we know that this drug is helpful for a particular disease, but we do not know how it acts. So there can be a pharmacodynamic study, or we do not know how this drug is excreted. So there can be a pharmacokinetic study on finding out, and then we, connect the dots and we get a better picture. Or one drug, for example, very common example is Viagra was discovered it was a side effect, that it caused erection in men because it was primarily cardiovascular drug to lower the blood pressure and ease the chest pain and stuff. So, we need to study that side effect in detail in order to implement it. And many new drugs have off-label uses. For example, some of the medications which physicians know from the experience with patients who are using it, that it's beneficial in some other category as well, not only in what it is intended for. But since it's not approved for that indication, so there's a gap in the knowledge. So what they do is, it is not present in literature review, and do that analysis of the existing database. Then we do a study, we conduct a study, we can do a randomized control trial. We have two groups of patients, one receiving the drug, one receiving standard therapy or placebo. And then we study the medication for that particular indication and see if it is, and then we generate more data which proves it, and then the gap is filled out. So literature reviewer tells us what exactly is out there and what needs to be done. Then we, depending on what kind of question we're asking, are we asking to see how across the different studies the results have been, for example, and the effect of COVID-19 on healthcare workers. So how it's been throughout, like in various countries, how people have experienced. So that would be a meta-analysis of so that the study design will be different. We'll be doing analysis of different, or if we want to find out like the causality, then the research randomized control trials are a good subset. But if you want to see what is the prevalence of a disease in a particular time, for example, how many hypertension patients, then we'll do a cross-sectional study to see that. So the design of study is appropriate to answer the research question. Could be like RCT, cohort study, case control, cross-sectional study, or meta-analysis. So when you are reading a protocol, analyzing it, so you have an idea, it's RCT, so what kind of things will be involved in that? It's a cross-section study, what is expectations? Then we see what is the study population and sample size, and that would be the important for recruitment, for what the inclusion exclusion criteria are for the population to be included in the study, and it should have enough. Sample size should be big enough to ensure the study has enough power. We examine the intervention, like what medication is being used. There's information brochure for that, and investigator brochure IB, and that will give us all the details of the medication, whatever has been out there up till now, up to today. So all existing side effects will be mentioned in there. What the dosages which people can tolerate, what are the how the ways of metabolizing, all that stuff. And we also need to know what the comparison group is getting. Is the comparison group getting a placebo? Are they getting a standard treatment? So the standard treatment and the placebo and the intervention, or the what's called the like the drug which is under investigation. So they have to be they have to be given at the same time. They should be in the same form, like the dosage method should be the same. Like if one is given IV, other has to be given IV, same way. So the dispensing schedule is also the same. So there's no bias, or there's no, it's like a double-blind study, so there's no bias. Then we need to find out like how are we going to define like that this particular like like the endpoints. For example, how will we know that what we're looking for, like that the question is answered or not? So the outcome should be clearly defined, and method of measuring them should be reliable and valid. So there should be endpoints defined. Like we're gonna, let's say, so that a new drug used for hypertension, is it better than the existing therapy? So the endpoint is that it lowers blood pressure more than the existing therapy. So that would be one of the endpoints. Second endpoint would be it will have like the ways it is excreted. So we will integrate pharmacokinetic studies. Or what are the like quality of life? People who are taking the drug, do they have any other comorbidities due to the drug? So are they able to perform well? Are they able to sleep, eat well? Are they able to do the activities of daily living? So that's another secondary outcome or exploratory measures. And then when we employ exploratory measures so that new studies can be done, which the data which comes out of them. There can be many secondary and tertiary endpoints, but our primary and secondary endpoints should be very well defined. And the how to measure those, like for example, what machines are going to be used for checking blood pressure, how much the reduction should be, in what posture will the blood pressure will be measured. So these have to be clearly defined, and then they'll be analyzed later on. The other aspect is these are important for coordinators for study population sample size. So if a coordinator gets a protocol, the first thing they're going to look at is the inclusion exclusion criteria on like uh, who I'm going to include in the study. So because they are the ones who do the consenting, then they need to understand the intervention groups also, because they need to know that what medication is being given to the patient and how it's going to be administered. They will be in charge of receiving the medication and keeping a log of it, dispensing, destruction, everything. Then the outcomes. So they will be getting the lab values like investigation. So there will be baseline lab values and investigation blood work done throughout the study to find out has there any changes, are there any biomarkers set, which is going to as per the outcome. Then data collection, very important part of the research coordinator's job is data collection, because they will collect all this data and upload to the sponsor website. So they need to understand what are the software used. Next is understand the statistical analysis, which is important part of the study, but very less frequently understood unless you're writing a research paper or you are the physician investigator. Very few people usually have to understand how this works. There's this very specialized team of people who write this part of the protocol, because it's without it, the whole validity of the protocol can go astray. We look for ethical considerations, because we need to have patients on the study um, who consent, and then the protocol is approved by the ethics board. So does it is the consent form in actually is simplified language? Is it simple enough for people to understand? It should be at a level of English, level of grade 7 or 8. And it does clearly explains the benefit of risk to participants. Does it tell them that it is a voluntary participation, and then you have to, you can say no anytime. Does it define any compensation to the participants, or is it giving excess amount of money just for participating? So that would be a bias. So next is evaluation of different biases. So how are you gonna randomize a patient when patients are included in the study? Are they going to be put into one of the two groups? How are they going to be put there? Is it going to be through a computer system or like IVRS system? So we have to remove all biases for randomized control trials, so the results to be valid. And then the publication of results and policies and stuff like that. So when you read a protocol, you should look for these 12 points, then you have a better idea on how to go about the protocol. The thing is, coordinators need to prepare some charts. They need to prepare a timeline. For example, when the patient is consented, when the patient came for the baseline visit, and what are the follow-up visits, what the schedule is like. And then they need to create the consent forms. They need to go through all the case report forms for data collection. They have to get training on the, they have to get training on the software websites. They're going to enter the data. So this is all provided by the sponsor. And then they have to create and maintain a regulatory binder in which they put all the documentation, so that if anybody comes for inspection, they can actually have evidence to present that this is a valid trial. It's been approved by the regulatory authorities like Health Canada or FDA. It is approved by the Research Ethics Board. All the documentation is in place, and all the consent forms of patients are there, so that they know there are actual people there out there who are actually doing research on. Next, we come to the steps to write a clinical research protocol. Like, for example, you are a research scientist and you want to write a new protocol on. So what are the various things you're going to be looking for? So First and foremost, the title of the protocol or the, your research question or the hypothesis. That's the most important part because that's going to direct how the whole protocol is going to be about. Um, it should be a clear title. We'll discuss each of these in more detail as we go along. A clear, concise, it gives you as much information as possible. Possibly includes the type of study, includes but the main endpoint is and main research question is about. Then the next section is the background and rationale. Like in the real protocol, before the title comes, there is a like the investigator signature page, the introduction, like all the people on the committee, and there's a like the index of all the items, and then after that there comes this section, so the abstract for the whole protocol. Abstract contains a little bit of information in summary of all these sections, and then all these sections start and then give information in detail. So we have background and rationale. So to provide a brief review of the existing literature, for example, I just mentioned that there has to be a gap in the science which the protocol is filling, and or examining something completely new. So we with the references from the journals and existing research, we try to explain that why we are doing the study, what is a reason that we are looking for a new medication which will cause less morbidity and more effective or faster acting or shorter duration or an easier route of administration, like it's given orally versus IV, which is more cumbersome and it requires more professional expertise. Or it is cheaper, or easily accessible, or it is available from like a natural products without no artificial chemicals and stuff like that. And the also background, and then the research question and objectives. So what are you trying to achieve? We say that this medication is as good as the existing medication or better, or stuff like that. Then we do describe the study design. So what kind of trial it is? Is it a double blind, placebo controlled phase 2 or phase 3, study, or it is case control study, or it's a cross-sectional study. Then we discuss the study population and sample size. So um, it will define the population and inclusion exclusion criteria in great detail, because there are lots of confusion about who to include. And if you have a wrong participants, like people who do not meet the criteria, then the validity of the study goes astray. So it's very important. And this is very important section for the coordinators, because the first thing they look at is who's going to be included in the study, and what exactly they need, how to screen that patient, what are the various things they're going to need to get that patient in the study. Then we look at intervention and comparison, that what are the procedures, same as analyzing a protocol, but in this, when you're writing it, you involve for not one person is going to do all this stuff. So this section might be written by a pharmacist, like the investigator brochure and other sections. The inclusion criteria might be written by a coordinator, and the study design, research question objectives can be written by a scientist, like whose main job is to explore what's out there, to go out in the field and form new hypotheses, or by a physician investigator who sees that I see patients in the clinic and I need to find out what is going on. I need to find out how this disease works, what is the natural history of disease. For example, COVID-19, how it progressed. There were studies done, they will have like tissue samples. They were infecting the tissue samples with COVID to see what his effect is on the various organ systems, miniature organ systems, and they say it destroys everything. Then they studied microscopic level to see it was causing vasculitis, it was causing heart attacks causing, brain symptoms, stroke, and all this stuff. So it's natural history of disease. So the scientists, can study this. So research scientists will write this part of the protocol. Then data collection and management could be written by data scientists or data managers, because they are the one involved in collecting data from different sites. So you have to define how to collect data. So what kind of case report forms you're going to be using. So you have to give a sample, a blank forms, and when you collect information, how are you going to store it? How are you going to, is it going to be stored on the server? It's going to be stored on the on site, and how will it be transferred to the site sponsor, and how we're going to analyze it in the end. So outcome measures, the secondary, primary, secondary outcomes. So this could be written by the lab director or the investigation. So you have to find out which investigations are required, to find out that there are changes as we expected. So we compare the changes between two interventions, and then see this intervention more effective. Let's say one intervention reduces the blood glucose level more than the other. So that's a better drug for diabetes, or it's more useful in shorter amounts, and so less chances of side effects. If something can be given in small amounts, is more useful. Then ethical considerations. This could be driven by ethics coordinator in the protocol team, and they will write down the ICF and consent, all the consent documents, all the documents should go to R&D. Then potential bias and limitations. This is usually dealt with by the statisticians. Timeline and work plan. The managers, they clinical trial managers, they see the feasibility, different stages. So it will have to be passed by clinical trial manager, whose main job is to run the operations of the study. So they know what it is like, to be working on the study, what kind of hurdles can come through, and then they will check the feasibility of this happening or not. And then in the end, you cite all your sources, you collect all this information from, so that it is a valid information you can always check. We will go and see at the real protocol. There is an example here with different sections in the end, and I need to change screen and do that, but you get the idea. You guys can raise your questions write in the question in the chat, or you can stop me and we can answer it, as we go along. Okay, so for the title of the protocol, it should be clear and concise. It should be informative. It should reflect the research question. It should use active, not a passive language. It should be specific and detailed. Avoid any sensationalism, and use keywords. For example, study protocol of a phase 2 clinical trial. It's defining the phase. Examining oxaliplatin, mentioning the intervention for treatment of HER2 negative advanced recurrent gastric cancer. So defining in what patient population, previously untreated with chemotherapy. So we are seeing what effect this particular intervention is going to have on very specific group of patients. So this is a good title of protocol. Then in background and rationale, we discuss literature review, we define the problem, define the justification for the research, why are we doing this research, what public benefit it brings. That's a huge question in ethics when you submit. So they always ask, how is it going to benefit the society? How is it going to benefit the patient population? Or then we give the study rationale. Then we define the research question hypothesis and its potential impact. There's a form we have to fill out when we submit application to research ethics boards. That is the the impact on various departments, like this protocol when implemented. So all the departments to have, we need to have signature of, for example, the lab department, what investigations will be performed for the study, or the pharmacy department, and the cardiology technician for ECG and stuff. So, or radiology department. So, what impact is that when while it's being implemented in the clinical setting, and potential impact of the intervention when it's approved for marketing in the population. Research question. This is the central question that your study seeks to answer. It should be clear, concise, and directly related to the problem identified in your background and rationale. For example, and then there's like in adults with type 2 diabetes, does metformin compared to placebo reduce blood sugar levels over a period of 6 months? So it's a very clear-cut question. We're comparing very specific medication in a very specific patient group, because people have written protocol like questions like, is this drug helpful in patients of diabetes? Like a novice would write like that, but you have to understand that you have to be specific. You have to answer a gap in the science. You have to answer a gap, it's out, what is inexisting, what's out there. Then the objective is specific goals you aim to achieve with your study. They should be specific, measurable, achievable, relevant, and time bound. So, for example, they assess the effect of metformin on blood sugar levels in adults with type 2 diabetes over a period of 6 months. So specific, measurable, so over what time, achievable, relevant, and time bound. So when you incorporate all this, it gives clarity to everybody on what should be doing to achieve that result. So the more clear the objective is, the more clear clarity is in the clinical operations team, and then they can work towards getting that goal achieved. Research design, as I mentioned, so there are different types of design. The experimental design is the randomized controlled trials. They are the gold standard because they answer the question, is this medication treatment is effective or not? And there are usually two groups. One is the intervention and one is the control group. Then there are observational designs, like cross-sectional studies to find the prevalence of a disease, or cohort studies, prospective, retrospective, and to go back and see, for example, case control studies were done to see these two effects of smoking in adults. So lung cancer is caused by smoking, so the first one was retrospective study. Then there are qualitative designs. For example, you're taking an interview of a patient. For example, there was a study done to find out the effect of intergenerational trauma, and how that affects, like how it increases the chances of suicide in Aboriginal population. So Aboriginal population in Canada have been abused for many years. They're first generation, second generation. Kids were taken into boarding schools, and they were all sort of maltreatment. So how does that intergenerational trauma has affected them, and how it is increasing the risk of suicide in the population? So this is through qualitative interviews. We just talk to and then we try to find causality. So it's a different type of study design. Or there could be mixed methods in which there are, both interviews and like interviews for quality of life, and then so an intervention and then the quality of life study. So they combine together. This is also, the next part is a study population and sample size. So it is important to define your population in terms of specific inclusion and exclusion criteria. And as I mentioned earlier, this is like in what age, gender, diagnosis is going to be including in the particular protocol, and how you exclude those patients, like for example, patients with severe comorbid diseases, like which can deteriorate when they are on the study. Second is pregnant women, or people who might get pregnant will be excluded from the study, or somebody you know, do not have the mental capacity to give consent to the patient, that can be excluded from the study. Or we want a specific group, like we want people with hypertension from like 150 to 190. We do not want people with very high, like more than 150 systolic, or less than, one, so like more than 190 systolic, or less than 150 systolic. So we want specific range of, people with specific blood pressure readings. So the others could be exclusion criteria. Um, the next is the, the statistical analysis. This section is written by the statisticians. So they have like different calculations to find out the power of the study. This is the most important section in the statistical analysis. That the probability that the study will detect an effect if there is one. So if you do not include that properly, the whole study can go astray. Like it's no use spending billions of dollars on. This is a very important part of the protocol. Then we look at the intervention and comparison groups. There's a section which defines what medication are going to be used, the nature of the intervention. Like, is it a drug? Is it a medical device? Is it a natural health product? And what is the dose, frequency of administration, duration of treatment? Is this going to be a crossover study in which one person gets first the intervention, then the control, and vice versa? Or is it a double crossover study? Or why this intervention is chosen? There has to be clear detail about what's out there in the investigator brochure. The procedure on how it will be administered, by whom can a coordinator administer this medication, or do you need a specialized nurse to administer this? Any like follow up, and then how are you gonna document the adverse effects? So that's a very important section of the protocol to define the safety. So what investigation will you do if you find the side effects? What are the expected side effects? What are the unexpected side effects? And how to document those, how to report those side effects to the sponsor so that they can submit those to the regulatory agencies. Then we have the exactly same mention for comparison group, even though it is already approved and marketed, but we have to treat it as a intervention, as the investigation product, as the IP, because we cannot treat it differently. The patient will come to know that I am getting this and the other person is getting the real drug. So you have to mask it. There should be, even from the taste perspective, it should be, if the IP has no taste, the comparison group should not have any taste. If IP is given IV, the control has to be given IV. And you have to mention, now there's an ethical consideration here that you cannot use a placebo in case there's a standard treatment available for especially people who are gonna go rapidly down for fatal diseases like cancer. Like, you cannot use placebos in cancer. So you have to use standard medication. There was a study done in syphilis, Tuskegee study, very famous study, and it was a lot of turmoil on ethical considerations. One group was given penicillin when it was discovered. Other group was still given, water injections, and that group deteriorated, and many of them died. And so, from human safety, dignity, and confidentiality is number one. If any of the studies breaches that, then you cannot publish it, you cannot use the data, you're liable to prosecution, you could be fined, even jail time. So ethics is very, very important. You have to realize that you're doing this on a fellow human being. So you have to understand that before, when you write a protocol, so they should not, they should be properly explained what are the risks of being in the study, and they should willingly participate. and they should not be coerced into participation by giving thousands of dollars or some other benefits or gift cards or something like that. So most of the people who participate in the study usually like have ongoing condition which there is no other treatment available, so it's their benefit if that treatment is effective, which most of the times it is. They get for free, they get extended care, they get like closely looked at. Their investigations like performed more frequently. They have easy access to the doctor appointments and stuff. So this is all important for medical considerations. And then randomization procedure. So it has to be mentioned in the protocol, like how you're going to assign the participant to each arm. When you put the eligibility criteria in the computer decides which one gets it. So they're randomized. So there's a the treatment allocation is provided to the site, and earlier it used to be phone by phone, but now things have changed and there are various softwares you can use, IVR software you can use. Then, as I mentioned earlier, data collection. What data is collected? Now there's a huge like flaw in data collection. People try to collect everything. It is useless and does not conform with the research question. Only collect data which is needed. Many people want to have like extended health CRFs with details and details of which I'm never going to analyze, and that is expensive, time consuming, and it decreases the morale of the participants. They have to sit for long hours when they're coming for the studies. So it is important to collect very thoughtfully and carefully design that I need to find the answer to this question, and for that I only need this data. If the like let's say information about what time you sleep or how many hours of sleep you get at night is not required, then don't put that in the form. Or if how many meals a day you're going to take, if that is not required, don't put that in the form. Don't try to collect everything. Also, you have to define, so the CRFs are based on very intelligent data collection strategies defined by the data scientists and the data managers. Then we have data collection instruments. How are you going to do it? Are you going to do it electronically on paper forms? Is it questionnaires, lab tests, or physical examinations? Mostly the electronic patient records are being used to collect data, and that's the source documents. We have lab investigation and stuff. What are the procedure? When it will happen, and how often will be collected? So the schedule of visits have to be defined. Will be collected on baseline, first month, 3 months, 6 months, 9 months, and 1 year. And then we do all these repeated tests. If there is an adverse event, then we do all these battery of tests and we report that. And and how will we manage? Is it recorded on a computer electronically? Are the patients going to do their patient reported outcomes like questionnaires on a tablet, or are they going to talk to the coordinator and he's going to record, document those things? So these have to be mentioned and in the protocol approved by the research ethics board, approved by the regulatory agencies, saying that it's all good. Now there's a new thing in data management that all the equipment which is used for data management has to be calibrated and validated. That all the computer systems, so it started recently, it was not there before, to bring standard, like universality in the like data collection across all sites. Then the quality assurance procedures, QA and QC management. So quality assurance is the process, the plan of how we achieve quality, whether it is data or whether it is a manufacturing a product. There is a data quality assurance QA in the manufacture of the intervention, or the manufacture of the investigation product or the drug we're going to be using. That is important. So this has to be mentioned. Also in the protocol, we mentioned the monitoring plan on how people are going to come. That's part of QA QC. So monitors will come and visit the site and they will see if the study is going on as planned, if there are any deviations, if the real patients, real data, I know what the site is like. So um, that monitoring plan is a mandatory thing for now to be included in the protocol and sections. Defining the outcome measures, for example, the endpoints as I mentioned earlier. What are we like? What is the important outcome that will answer the main research question? So if the blood pressure goes down as expected, then we reach the target. If the blood glucose level goes down as expected, then we reach the target. If the disease alleviates the drug alleviates the pain or extends the life in a cancer patient, then we achieve the outcome. So very clearly defined. Then we can add secondary measures, measure like safety, adverse events. Now, all this writing, all this stuff needs the knowledge and experience of knowing what's out there already. That's why research scientists are the best people to write this, because they interact with this on a daily basis, with these patients on a daily basis. They know what are all these measures they are looking forward to, what are the safety implications, and or PhDs, or like, so they are the people who can actually tell what exactly is required in this part of the section. I mentioned this earlier, so you will be presenting your in the research paper the findings. So for that you need to have descriptive statistics when you're writing the paper, inferential statistics, and also what to mention how you handle the missing data, plan for dealing with missing data. I'm going to analyze subgroups, or there's going to interim analysis. The data safety monitoring boards usually do interim analysis. So this is, for example, studies done for 3 years, and they do analysis in 1.5 year, and they see that the amount of the patient population who was employed have achieved the target outcome, like whatever they were looking for, the research question answered, and answered in as many people are good enough people to prove that it is effective and it has a power, then they stop the study there. They don't need to conduct the rest of the study. They don't need to go all the way to 3 years, because it saves time, saves money, which is huge, like billions of dollars, and early marketing, which gives them the marketing of product, and which gets them the purchasing power much earlier. So every year counts, because they have a 12-year, like they have like a 20-year period, 12 years research. So 8 years is what they get the exclusivity to market the product. So they get exclusivity for longer time. They can make more money. So interim analysis is important. Also, interim analysis is good. Like if they do and it's not working out, and a lot of people are having side effects, they close the study because it's not good. It's not going to answer the question. So data safety monitoring boards are independent committees which look at the results of look at the data, and then they assess and see if like this protocol should continue, and they give just a one-liner report every year. This study should continue, or this study should not continue, then they give data report. But most of the time it's we have reviewed and then this study should continue. That's it. So they will look at everything and then say, okay, we can continue the study and complete it. Data management, and it also includes like software. What software are you going to use for, so there are like different training software for the patient, for the coordinator sites, and different PRO, patient reported outcome software for tablets for the patients. There are like data collection, so all these things can use. Then, as I mentioned earlier, the ethics board approval is a must, and you will ensure there's informed consent, there's risk benefit assessment, patient privacy confidentiality is taken care of. There is HIPAA and PIPEDA regulations here in Canada, and there can be $50,000 fine on individual that breaks the confidentiality. and $250,000 fine on the institution, which breaks the confidentiality of the patient. So it's very important that it's their data is kept as it is planned under lock key, under password, and not revealed to anybody else unnecessarily. Okay, we're nearing the end of presentation. Then what are the potential bias and limitations? I discussed randomization already. Then there's blinding. So this removes bias because nobody knows who's getting what. I don't know what patient is getting. Patient doesn't know what he's getting. So there's no way any preferential treatment. And using standardized instruments, for example, calibrated instruments for data collection reduces bias, and we use inclusion exclusion criteria to remove bias. Then adhering to protocol, like this is important. And I also do independent data monitoring, like data safety monitoring boards or DSMBs, as you call it. Then feasibility, like the operation managers will look at the protocol, say, is this going to work out in real world or no? Like, do we have enough resources? The site will look at it and say, is it feasible for my site? Do I have enough resources? Do I have enough staff to be able to do this? Do I have the lab facilities to take all the samples? Do I have enough patient population? First, the most important, the most challenging aspect. Am I going to be able to recruit patients in the study or not? Do we have the equipment, and do we have, so they check everything. First of all, protocol, from the protocol writing perspective, if the protocol is feasible, it's going to be feasible on the site or not, to what exactly do they need in order to conduct the study. And then the site has a feasibility, because every site is different. They will see, okay, this is protocol I can do, this one I cannot do here because this needs specialized equipment or specialized staff. And you have to define a timeline and work plan. See, define all the tasks. So there's a Gantt chart the operations team can create. They will assign tasks to different people, the timelines, and then their critical tasks need to be completed in order to have the next one being done, and then we finally achieve the product. We assign responsibilities. This is usually not in the protocol. This is usually during the analysis, and the sites do it on their own. For example, when the site receives protocol, they will create a timeline and work plan, and then assign the tasks to different staff members. But it is good to have an idea when you're writing protocol, what would look like from the site perspective. So that will also determine the feasibility of the study. Is it going to be, helpful for that or not? Then how are you going to, publish the results? Many of the patients have asked me who are in the study, I'm not going to get the results. Will I ever know? Was I on the intervention group or on the placebo group? I said, yeah, for sure. You have the right to your the results. Once a paper is published, I'll send you a call. I'll give you a call and I will send you the paper copy, of the published paper. But usually it's so long, after studies complete 3 years, just complete the study, then another, it gets logged, data logged, get cleaned for a year, then another 2-3 years to get approved. So by the time people forget, most of them, 4 or 5 years, by the time it's published, they forget. And no one has ever gotten back to me 5 years later, say, oh, by the way, what happened with that study I was participating in? Do you have the results now? Never happened. Also, you need to, choose the timing of your publication, what resources you will need for writing, because you need medical writers to write this information, to do research paper and stuff. And then in the end mention the references from where you got the information from. It's very important. Without this, the whole protocol is invalid if you don't write the references, because anybody can make stuff up. So there has to be general references in a specific format, like AMA style, APA style, or ICMJE style. So you mentioned that it's give validity to your protocol, you're robust, and people can go back. It also helps find new questions, new answers. If I want to study something, like a gap, I'll find to do further analysis. So, okay, awesome. So we done 1 hour. I'm open the talk to presentation. If you like to ask any questions, anything you didn't understand, we can go back and discuss. And any comments and feedback, if you like the presentation, any suggestions for improvement and stuff like that.
Hi, Sunit. I just wanted to ask, are there any particular softwares which companies are using for designing the protocol, or is it done manually with the help of like the basic computer applications?
Um, I think the word is Microsoft Word is the most commonly used. But as I mentioned, different sections of the protocol are written by different people. So statistical analysis, they employ those softwares, which I'm not aware what exactly they're using. And then people do like a journal search for the final science rational background.
Mm.
So they have special softwares to do journal search,
Okay.
their databases, PubMed, Google Scholar and stuff like that. So they are used, and then data scientists are going to use softwares which are going to be actually used in the study, like when implementing the study. So they will do the testing and trial. So that is these are the different things you can be using. I don't think so. There's one software you to write a whole protocol. You can integrate. There are also electronic trial master files for collecting data. They're like a pharmacovigilance software for collecting adverse events. So
Mm.
these are incorporated into the different subsections of the protocol as and when needed. But I don't think so. There's one particular software in which you can just use it to design the. You have templates, like there's a protocol template which has all the different subsection and the questions which I
Mm.
came across, and you can fill in the template. Okay, this section, these questions are being answered. And
Okay.
then you fill out all these questions, your protocol is there. So that's kind of.
Okay, got it. Thank you. It was very nice and crisp session.
Okay, awesome. I'm happy you guys are, I mean, anytime you have any questions, so please find my other group,
You
and we will discuss how to help you with your finding your
will
career.
send me the link